So hello guys, welcome to my YouTube channel Med School with Shubham. I am Shubham, a third professional MBBS student, and I shall be teaching you anatomy. I have started a new series on embryology on my YouTube channel, in which I have completed the first week of development and the second week of development, and also started the third week of development. In the third week of development, we have completed the process of gastrulation and also completed the formation of the notochord. This is the third part of the third week of development, in which we will be learning about the process which is called as neurulation. So what exactly is neurulation? So first write the definition of neurulation. So neurulation is the process of formation of the neural plate, neural folds, and the closure of the neural folds, to form the neural tube. So this is called as neurulation. So let's read once again. So what actually is neurulation? So neurulation is defined as the process of formation of the neural plate, neural folds, and the closure of these neural folds to form the neural tube is called as the neurulation. So now let's learn the actual process of neurulation now. Okay. The mechanism. Okay. The mechanism of the neurulation. So what will happen? Guys, if you have not watched my previous lectures, I request you please go on my YouTube channel and also watch my previous lectures on embryology, so that all your concepts of embryology will be crystal clear till this point, and it will be very easy for you to learn the future things. So this is the ectoderm, this is the endoderm, and this is the intraembryonic mesoderm or the mesoderm. And in the lower part towards the endoderm, we have a structure, newly formed structure, which is called as the notochord. I hope you are right, you have named it. So this is the ectoderm, this is the endoderm, this structure is the notochord, and this is the mesoderm. Okay, so now what happens? The formation of the notochord stimulates the surface ectoderm cells. The formation of the notochord stimulates the surface ectoderm cells. This is the notochord. So when this notochord will be formed, the surface ectoderm cells will get stimulated and they will become thick. They will become thick as compared to other cells. Okay, in this way. Okay, so what will happen? The formation of notochord will stimulate the surface ectoderm cells to become thick, and this is called as, and this thickening is now called as the neural plate. Okay, so in these three things, that is neural plate, neural fold, and the closure of the neural fold, we saw the first thing, that is formation of the neural plate. So this is the neural plate. So what is neural plate? The notochord is formed, and now the formation of notochord will stimulate the surface ectoderm cells to become thick, and this thickening is called, these thickened ectoderm cells are called as neural plate or neural ectoderm. It is also called as neural ectoderm or neural ectoderm. Okay, now let's see what happens further. Now in the next step, these will now move upward in this direction and they will form a fold like structure in this manner. So let's draw that. This is the ectoderm. Okay, so now this is formed, and below that, or instead I should draw this diagram first. Wait. Okay. So this is the ectoderm. Okay, and beneath that we have this endoderm, and in middle the mesoderm. Now what will happen? This pit will go more deeper now in this manner, and now these ends will become in this manner. So it will form a structure in this manner. And in the lower part we have this notochord here. Okay, now let's name the structures. So this is neural fold because it has formed a fold like structure, therefore it is called as neural fold, and this thing, it is called as neural groove. Neural groove. This is the neural plate, then we got the neural fold, and now we got the neural groove. And one more interesting thing, that on the ends of these neural folds, means on this structure, on the ends of this neural groove, okay, neural folds, the cells gets differentiated and they get modified. The ends of this neural fold, means this is the entire neural fold, this is also a neural fold. So the cells which are on the tip of this neural fold, they get modified and they get differentiated, and now these cells are called as the neural crest cells. Neural crest cells. Okay, so now till this point we got the neural plate, then we got the neural fold, then we got the neural groove, and now we got the neural crest cells. Okay, so now let's move forward. Now, so what actually is this? Assume that this is the entire embryonic plate. Okay, let me draw in this manner, in this diagram. Copy. Paste. Okay. Now we saw the formation of the notochord in this way. Here was the notochord, that is from the prochord, from the primitive node till the prochordal plate. Now this is the notochord. When the notochord is formed, the surface ectoderm cells will get activated. This is the notochord. Assume that this is the notochord. This is the notochord. Now, because of the formation of the notochord, the surface ectoderm cells, means which is above to the notochord, they will get activated in this manner, in this line. So wherever there is notochord, the ectoderm which is above to the notochord, they will get activated, and due to the formation of the notochord, the neural fold will come in this manner. It will formed in this manner all over. So you can see the formation of the neural fold in this way. I hope you can appreciate this thing. I can't show you in real now, but all I can try is this three-dimensional drawing that I can do. Okay, so okay, I'll draw that. Okay, see, I hope you can appreciate this drawing. The surface, assume that this is the surface ectoderm cell, ectoderm layer, and now because of this formation of this notochord, wherever there is notochord, the ectoderm which is above to the notochord, it has elevated upward. Wherever there is notochord, assume this is the embryonic plate, embryonic disc, or the embryonic plate. Now this is the notochord. Assume this is the notochord. Okay, this is the prochordal plate, and this is the prochordal plate. This is the primitive node. Now, wherever there is notochord, the page will now come upward in this manner, Okay, so it will look something like this. See, in this manner. Assume that this is the page. So a groove will be formed. Okay. So a groove will be formed in this center, and the fold will, I hope you can appreciate. Assume that this is a fold and this is the groove in the center. Okay. So similar I have drawn here. So this is the neural groove, and this thing is the neural fold. This is the neural fold, and on the edges of the neural fold, means this thing, you have the neural crest cells. I hope this is very clear to you. All I can show you is this diagram only. So try to understand this thing. All these are just a kind of three-dimensional things, but I can't show you more properly as compared to this. So if you have any doubts, you can directly contact me. I will explain you once again. But I hope you have understand. I hope you have understood now. So this is the neural fold, and this is the neural groove. The entire thing, the entire ectoderm, it gets elevated, and a neural groove can be seen from the upward. So this is the neural groove. Okay. Now next thing, this neural fold has to be closed now. We have to form a tube. So tube is all closed like a pipe thing. Okay. So the pipe has to be closed. So now these things, it closes now. That is, these neural folds, it gets closed. See, this neural fold will now get closed, and you will get a circular neural tube now. Now this closure, and this is what it is written, that is closure of the neural folds. So this closure of the neural folds, it takes place first in the cranial region, means in this neck region. This is the entire neck. So in the posterior aspect of the neck, that is the cranial region, in the cervical region, sorry. It will first take place in the cervical region. Then assume that this is the two neural folds. First the joining closure of the neural fold will take place in the cervical region. Now it will move in the cranial region. I mean, it will fold in the cranial, it will close in the cranial region, and then it will fold in the caudal region. I hope this is clear. First, the closing takes place in the cranial region, I mean cervical region, then it will close in the cranial region, and then it will close in the caudal region. Okay. Now, closing takes place. Closing first takes place in the cervical region. First takes place in the cervical region, and then it becomes bidirectional. That it will go in the cranial region also, it will go in the caudal region also. Bidirectional means what? First it will close in this region, okay, in this region, and then it will move closing in this direction also, and then it will move in this direction, because this is the cervical region and this is the cranial region. So after closing in the cervical region, it will go craniocaudally, means in towards the cranial end and towards the caudal end. Okay. Now, so the fusion will take place in the bidirectional, that is craniocaudal. that is craniocaudal direction. Okay. Now after the formation or after the fusion, what we can see now is this is the ectoderm, this is the endoderm, and in the center we have the neural tube. Now after folding it will form a tube within, so this is the neural tube, and in the lower side we have this notochord. Okay. Now one thing that comes over here, where are the neural crest cells? After folding of the neural folds, where will the neural crest cells go? So one thing happens that during the folding of the, see, these were the neural crest cells. So after folding of these two things, where will these neural crest cells go? So during the fusion of the neural folds, the neural crest cells will get detached from these neural folds. During the closure, the neural crest cells will get detached from the folds, and then it will come towards the lateral side of the neural tube. So let me name them now. This is the neural tube, and here we have the neural crest cells which got detached from the neural folds at the time of fusion of the neural folds. Okay, so this was all about the neurulation, and last thing that I will mention is the clinical anatomy. So we saw the fusion. Oh sorry, I forgot one thing. After the formation of the neural tube, now assume that this is the neural tube. So this neural tube is hollow from inside. This thing is hollow. Okay. So when this neural tube is hollow, let me show you one thing for that. Okay. Assume that this pen is a neural tube, or this one is better. Okay. Assume that this pen is a neural tube. Now it is hollow from middle, in the middle part. I hope you can see here. It is hollow in the middle part. Now this neural tube will be having two ends. As you can see here, this is one end and this is another end. Similarly, this neural tube will be also having two ends. That is, one is the cranial end that is towards the head, and other is the caudal end that is towards the tail. So after fusion, we have two neuropores. These ends are called as neuropores. Pores means opening, and opening of the neural tube. Therefore, they are named as neuropores. We have cranial neuropore, and we have the caudal neuropore. Okay. Both of these, cranial and the caudal neuropore, has also to be fused now. Both of these will also now get fused. They will get sealed off like this. Assume that this is the now this cranial neuropore got sealed off, and from back side also the caudal neuropore will now get sealed off. So the cranial neuropore will get sealed off on the 25th day of development. The cranial neuropore will get sealed off on the 25th day of the development, and the caudal neuropore will get sealed off on the 27th day. On the 27th day of the development, the cranial neuropore will get sealed off or will get closed. It will get closed. These neuropores will get closed on the 25th and the 27th day. Now here comes the clinical anatomy. That is, if these neuropores doesn't get sealed off, or if these neuropores doesn't get closed, then what will happen? So if the cranial neuropore doesn't get closed, then we will get a condition which is called as anencephaly. Anencephaly. And if the caudal neuropore doesn't get closed, then this will cause a condition which is called as the spina bifida. Anencephaly and spina bifida. These are two conditions which will occur when the cranial neuropore is not closed on the when the cranial neuropore is not yet closed, then it causes anencephaly, and if the caudal neuropore is not closed, it causes spina bifida. I hope this is very clear to you guys. Let's revise everything once again. So this was the third part of the third week of development, in which we were learning neurulation. What is neurulation? Formation of the neural plate, neural fold, and the closure of the neural fold. Formation of the neural plate, neural fold, and the closure of the neural fold to form the neural tube. Okay, now let's see the mechanism. The formation of the notochord. The formation of the notochord will stimulate the surface ectoderm cells. Formation of notochord will stimulate the surface ectoderm cells to become thickened, and they are called, and now these thickened cells are called as neural plate or neural ectoderm. Now what will happen? A small bulge will start forming. A bulge is formed and it will be having a pit over here. It will थोड़ा सा bulge हो जाएगा और उसमें एक छोटा सा pit आ जाएगा. It will bulge first and then we will get a pit over there. Okay. Now it will go more deeper and it will go more upward. Now this upward thing is called as neural fold, and this deeper thing is called as neural groove. And at the tip of the neural fold, we will get specialized cells which are called as the neural crest cells. Okay. Now this it will be longitudinally all over wherever the notochord is present. Means all over this neural fold is formed. All over this neural fold is formed. Now the closing of this neural fold will take place. First it will take place in the cervical region, that is in the neck region. This is the cervical region, neck region. Then it will go towards the cranial region and towards the caudal region. Then it will be bidirectional, that is the craniocaudal direction will take place. Closing of the neural tube. Then after fusion we will get two neuropores, that is the cranial neuropore and the caudal neuropore. What is a neuropore? The opening of the two openings of the neural tube. One towards the cranial end is called as the cranial neuropore, and the one towards the caudal end is called as the caudal neuropore. Those are called as the cranial and the caudal neuropore. These neuropores are to be closed. The cranial will close on the 25th day and the caudal will close on the 27th day of the development. If the cranial neuropore doesn't close, then it will cause a condition which is called as anencephaly, and if the caudal neuropore doesn't close, then it will cause a condition which is called as spina bifida. One more thing, during the closure of the neural folds, the neural crest cells will get separated and then they will migrate towards the lateral side of the neural tube. I hope this is very clear to you guys. I have explained you in the most simplified manner as I can. Even then, if you have any doubts, please don't hesitate to message me or direct DM me on my Instagram handle. I will be very happy to answer you and also clear your doubts. Also, you can also leave your doubts in the comment section where I can address your doubts and clear it over there also. Thank you guys. Please like my video, share my channel, and also subscribe to my YouTube channel, guys. Thank you so much, guys.